Okay. Should we start?
Yes.
Just say so.
Yes.
Can go?
Go.
All right. Good morning, afternoon, everyone. We have next iteration of our myeloid network. We have a very interesting presentation from Nan Zhang. He's young, but already accomplished investigator, assistant professor at Wistar Institute and University of Pennsylvania. Nan spent several years as a postdoc in Glenn Randolph Lab at WashU, and work on myeloid cells for quite a long time now. And recently he did quite exciting research and publications, resultant publications. He's mostly focused on macrophages in ovarian cancer. I guess his focus on his presentations will be around it. Nan, it is a really great pleasure to have you here today, and we're looking forward to your presentation. And as always, please put your questions to the chat, and we will make sure that all the questions will be asked and hopefully answered by Nan at the end of the presentation. Thank you.
All right, thank you so much, Dimitri, for the introduction. Thank you again. Thank you to Myeloid Network for inviting me. As I mentioned, it's such an honor to be here. To the trainees in the audience, I was still a postdoc when this thing started during COVID, and it was always my pleasure to come to this meeting, and from the very beginning. And I'm honestly very flattered to be here. So today I want to talk about. You guys can see my slides, okay? I assume. I think it's fine. Then let me minimize this. So it's about ovarian cancer, as Dimitri mentioned. It's kind of my new favorite topic now. And how do we use myeloid cells, leverage their activation in metastatic ovarian cancer? And as Dimitri mentioned, we're at Wistar Building Institute, which is located essentially physically at the heart of University of Pennsylvania in Philadelphia. For people who don't know this is important. All right, and then the people who did the work, I really appreciate everything they did, especially two talented postdocs, Brenna and Tidal, who essentially started the lab with me together with Brian, who's the first rotation student who started the rotation when my lab has nothing. And then I really appreciate them and the work I'll be presenting is from the two postdocs. And later on, as the lab grows, this is what the lab looks like right now. And this is a real photo that was taken from space, not photoshopped. As you can see, the glare. It was kind of an undergrad student. There was a project supported by Crunch Labs. It's really cool actually looking at this right now. All right, let's get into some science. So let's just get this straight out of the way. We work on ovarian cancer. I'm going to explain to you why we work on this and how I transitioned to ovarian cancer when I was a postdoc. So essentially some simple statistics for people who are not familiar with this cancer. Is one of the deadliest gynecological cancers and is now ranked as 6th leading cause of cancer death in women, as shown here. And is usually diagnosed in at late stage with late-stage disease, more than around 70% of patients, and their 5-year survival rate is really, really bad, 30%. And the reason why I started working on ovarian cancer is 95% of ovarian cancer or more, they always present with peritoneal metastasis, not other metastasis, not like a so-called blood hematological, hematogenous metastasis. So that's kind of unique to where I worked, in my previous times with Gwen at WashU. So that's why, focus on ovarian cancer. And it's really, really, bad, not really responsive like many solid tumors to immune checkpoint therapy. And I want to show this case and death numbers here just to highlight. You can see that you don't see the top 10 new cases in women, for ovarian cancer. However, it's number 6 in the estimated deaths in 2025, really highlighting this very high, around I think 60 to 70% so-called this death to case ratio or mortality to morbidity ratio, suggesting it's a urgent disease that's usually diagnosed at the metastasis stage. That's why we focus on metastatic ovarian cancer. All right, since it's peritoneal cavity, and let's go back in time and kind of just understand what myeloid cells, macrophages, neutrophils are in the cavity. So I'm going to spend a few slides just going over some of the old work I did, just make sure everybody's on the same page. When I was a postdoc, we really specifically focused on macrophages in the cavity, specifically in the peritoneal cavity, the abdominal cavity, shown here in humans. And there's also other cavities in the body, the so-called the pleural cavity, the pericardial cavity. They all have something called a cavity macrophages. Now the field kind of agreed on this. And then if you take them out, do like a PBS wash, we call it peritoneal lavage, and then get the cells out, you can see a lot of them are myeloid cells, macrophages in this case, in the steady state in mice. And I'll talk about human work briefly as well. So out of this macrophage gate, you can see there are mainly two subsets that are resident macrophages in the cavity. This is the so-called large macrophages. There's the smaller is small macrophages because physically they're smaller and physically they're larger. The large macs are the true resident macrophages. They're GATA6 dependent. They express a bunch of signature genes and markers. They are seeded prior to birth. They can proliferate locally, just like many tissue resident macrophages, as many of you are very familiar with already. And small macs from Gwen's lab, another postdoc discovered that it's coming from the monocyte, and then it's also microbiota dependent, IR4 dependent. So for today, for my research, we'll mostly focus on the large macs. All right, at the time when I was postdoc, we had this question, it was like around 2015, 2017. People in the field like Florent Genoux and others have discovered, Mima Raz and Gwen's lab have discovered macrophages are very different in different tissues, and there's tissue resident macrophages, monocyte-derived macrophages, as now we know as dogma. Back then, it was a new thing. Then different tissue macrophage has different microenvironmental cues or signals, as you can see listed here, 2015 review. In the same time, there were specific transcription factors like master transcription regulator that was discovered over here as well, specifically for this lineage of tissue resident macrophages. And we were focusing on peritoneal macrophages. Back then, we did sequencing, others did sequencing. They all know that these transcription factors that are governing so-called tissue-specific gene expression signature. However, back then, very few people were really, most people kind of assuming that's the case, that gene will translate to function, and then very few papers were really showing this definitively. And we were interested in, demonstrating this, especially using the peritoneal macrophage as a model in the very beginning. So the first thing to look at this is what is the microenvironment of these peritoneal macrophages. We were the first to develop this live imaging system using 2-photon. It doesn't have to be 2-photon, but we just use 2-photon and a live mouse, anesthetize the mouse, and just to image them using different reporter mice. Eventually, we landed on one model that was specifically labeling the cavity macrophages. I'm really proud of this image. It was taken December 23rd, 2016. When I was like first half a year when I was a postdoc, I was really happy to see this image right before Christmas. As you can see, the microenvironment for peritoneal macrophages is unique and very different from, like totally different, distinct from any other macrophage you can imagine in the body. They're highly dynamic, fluid, and this kind of raised a lot of questions: how they really function, can they really migrate? How do they really capture cancer cells, bacteria, stuff like that? And in the end, I'm just going to show you a key data of the paper, which is a little complicated, but I'm going to walk you through quickly. Hopefully, some of you or many of you have seen this data in the past, but basically we were trying to use infection as a model, or in the beginning is a sterile infection model using zymazan, which is a dead yeast, hollow yeast particle. And then what we found was what we were going after was so-called the macrophage disappearance reaction that was discovered 50, 70 years ago in the 50s, 60s. What essentially was whatever you inject an inflammatory agent into the peritoneal cavity, these macrophages will be just gone from analysis. You cannot wash them out anymore. Even using flow, using cell counting or whatever, you don't see them anymore. This is basically the counting the resident macrophages with specific markers, as we know now already. Their number dramatically dropped to less than 5%, like within hours, 3 to 5 hours after Zymosan injection. And it was known back then, somehow heparin, again, 60s, could rescue 100% of this, and it was consistent with that finding. And then, however, there was some baffling result that was generated a long time ago. We also reproduced, was using anticoagulation molecules. This is hirutin blocking thrombin, but there warfarin and others were used in the past. It's kind of seeing the same thing. Somehow there's a segregation between the phenotype of hirutin treated, heparin treated. Heparin is an anticoagulant. However, heparin can do many other things. And then in this paper, we showed, okay, it's not only coagulation that's doing this after activation, it's also integrin-mediated activation on macrophages, and then coagulation initiated by macrophages. We used different models in that paper. But this piece of data was essentially the keystone of the whole paper, holding everything together. I was really happy to see this in the beginning. And then the most beautiful data was if you block both clotting and macrophage adhesion, you reverse the macrophage disappearance reaction. And what's more beautiful is we found these so-called aggregates. Essentially, we're just calling them clots. They are white, and then they're real clots because we can stain fibrinogen, fibrin. The purple lines are here, the section staining, and they're macrophage in the middle, ICAM2. And neutrophil on outside is like 5 hours after the Zymosan injection. So we found these clots. And then more beautifully, which became the cover of that issue in 2019, is the fibrin mesh, which is a whole a whole mount staining of the clots, and you can see that the mesh is really trapping the purple Zymosan particles together with phagocytosis of macrophages. It's a very beautiful image, actually done by my colleague. And then we also show this true resident macrophages, GALA6 and ICAM2 double positive. All right, so that's just the important data we discovered in the past. Then the model for that part was, resident macrophage, different from many other macrophages in the body. They can secrete clotting factors, especially factor 5, and to initiate a clotting and cell adhesion, and to control bacteria, dissemination. Essentially, this is a little cartoon over here with bacteria, intrusion, invasion, and you can see these macrophages would aggregate, form clots in a clotting-dependent manner, and adhere to visceral organs, surface, mesothelium, momentum, to facilitate the trapping of bacteria. In this case, we use Zymosan, E. coli. So what's more interesting is what happens to these clots. And when I was trying to decide what I want to do in my own lab later in my career, later in my Ph.D. postdoc. So another lab from Spain, Dr. Carlos Ardovin, I guess. And Adrian, as the first author, followed up with what we found in the beginning in 2021, published in Immunity. Essentially, they were asking this question: okay, what happens to these clots? So I'm happily the data was reproduced, inject E. coli, and it found a bunch of fibrin, macrophage neutrophil aggregates adhere onto the visceral organ, and they followed longer than what I did, days, hours, and days. Eventually, these clots were cleared by monocyte-derived macrophages, which has a capacity to chew up those fibrin, like a fibrinolysis system. That was really beautiful. So not only that, and then later on, Dr. Paul Coops, together with Joel Zindel, they also did a 2-photon microscopy to image peritoneal macrophages. They extended the findings into a sterile inflammation model, which is laser-induced, peritoneal injury and peritoneal adhesion, which is really a big problem in clinics, post-surgery adhesion. What they also extended the finding with was they found this is a evolutionary very conserved process and the phenotype in these macrophages all the way back to sea urchins, these so-called chromocyte or basically macrophages or chromic cavities, eventually developing into multiple cavities in humans. I think there might be a mistake over here. These cavities are not really connected like this in humans or in mice. But the data is really showing in this steroid inflammation model, macrophage can aggregate just like infection model, and they also went further and identified interaction that's mediating this aggregation. They found essentially it's a physical an ionic ligand and binding positive negative charge that's mediating this aggregation. CD36, SRB1, those are the important molecules, and Marco, those are the ones that are important for this process. All right, so that's enough for like the stuff we discovered in the past. And then, however, back then we were wondering, when I was basically leaving the lab, we were wondering what happens to these macrophages in humans, and then do they look the same? Although Dr. Coops, they made a cartoon showing, yeah, they can do this. However, the transcriptional profile of human peritoneal macrophage or cavity macrophage wasn't really well done yet. So when I was doing this work, and COVID started, so I was working on both 2 human projects. I was really happy in the end. It worked out so well for me to learn human immunology. Later on, after I left, and then really talented postdoc in Gwen's lab, Ji Chang Han, I finished this work. Essentially, what we discovered in the beginning was, human and mouse peritoneal macrophages in so-called, in humans, we can only get so-called minimally. There's no true healthy donors, as we all know. These patients were undergoing hernia repair. They're non-cancerous, so we consider them as steady as possible. We also, in the end, after I left, they also did, pediatric patients who were undergoing minimal surgery, found the same thing. So we are considering that as a steady state resident macrophages. And what Jichang did was they overlapped, overlaid mouse and human peritoneal macrophages, clearly making the genes the same, and then making a UMAP together. As you can see, human and mouse macrophage in the peritoneal cavity just separate like so well. They don't look the same almost at all. In the beginning, we found that these GATA6 true resident macrophage in mice actually are pretty much absent. In flow, it's like single digit, like under 5%. And single cell, it's the same thing. These green, basically this tip that are highly enriched in mice, and we don't see them. However, these so-called monocyte-derived macrophages are some of the small peritoneal macrophages, like in mice, DCs, they're highly enriched in humans. We don't know why. That's why we did, in the beginning, we thought it was a developmental issue. That's why we did pediatric patients. We found the same thing. There was no enrichment of a mouse equivalent resin macrophage in humans. So we think it's an important finding. However, that being said, we still think the fluid-based capturing or coagulation, aggregation, a capacity of these macrophage probably remain the same, but we don't know how hard they're done, what's secreting factor 5, this factor, that coagulation factors. This is why, okay, later on, I guess we characterize more just to show you the differences between human and mice. As you can see, in humans, the GATA6 macrophage is just tiny. This p-selectin cell P is a GATA6 dependent gene, and TIM4 is a residence marker that we're working on right now as well. Then you don't see them at all. Being in mice, you see them all like basically all the large macs are like this. In contrast, these monocyte-derived markers, CCR2, MRC1, and then DC markers, DC1, DC2 markers, CD1C, CLEC9A, which is not showing here, they're all really high enriched in humans. This graph really highlights, the difference between GATA6 in GATA6 positive cells between human and mice. So after that, basically that was around time I was starting my lab in 2021. I was already transitioning to to ovarian cancer, then we were thinking when I was starting my lab, I was thinking, how do we leverage peritoneal macro activation to control ovarian cancer potentially? Um, um, so before getting into more details, I want to mention the most important problems of ovarian cancer. There are more than this, but these are the things I think that are really important that's making metastatic ovarian cancer so difficult to treat. One of the things is the so-called chemo-resistant cells, cancer cells that are floating in the ascites. You can see them all the time in all ovarian cancer patients. This paper from Nature Medicine in 2020 was just showing they are resistant to all the classical, chemotherapies. They're not shrinking. Maybe paclitaxel is doing something, but not enough. So is there a way to control this? And then secondly, there's immunosuppressive microenvironment, as we all know, almost in every single cancer, is myeloid cells, neutrophil macrophages, and regulatory T cell and fibroblasts. They're secreting a bunch of immune suppressive, molecules that are suppressing T cell killing. Another thing that's unique, or I guess pretty specific to the ovarian cancer field, is so-called a multi-compartmental metastasis, because peritoneal cavity hosts all these organs. They can go to the mesentery, the omentum, the liver, the diaphragm, all the membranes, and the fallopian tube and ovary. So as we all know as immunologists, this so-called tissue-resident signatures, tissue-resident microenvironment, is quite different among these organs. Are they important? Can we eliminate all of them at the same time, or not? So that's also an important question we were wondering. So the first thing that actually came to my mind was, as you may have predicted based on what I presented, can we leverage this macrophage activation phenotype in the peritoneal cavity, these fluid dependent clotting and adhesion signature, to remove these tumor cells in the ascites? Can we aggregate them and just take them out, just like in mice? Then it's an easy way to get rid of these cells surgically. So we did a few, we used a few ovarian cancer models, just showing a couple of them. And we picked beta glucan, which is similar to Zymazan, but it doesn't have the toll-like receptor 2 agonist, and activity. Also, it's beta glucan. Potentially, you can take from the grocery store, honestly. So it's easier, I guess, cleaner to study. Potentially having a more translational future than Zymazan. That's why we picked the beta glucan. But it's also dead yeast, essentially the same as Zymazan. And then what we did was just do 1 or 2 doses of injection of beta glucan during the progression of these cancers in the peritoneal cavity. I guess metastasis of these cancers. As you can see, 2 different models, and it holds true across board. You can see beta glucan treated mice. They have clear, no red acidic peritoneal fluid, which is a really good sign, suggesting beta glucan is clearing ovarian cancer cells. And we did confirm this with flow cytometry and others. It's a case. Then how this was done, and then I just want to give you basically the model, the cartoon of this part. And we end up using because cancer is chronic, and know there's differences. Eventually, for the convenience, we picked a short acute model to model this, to tease out the mechanism. It basically hours, and then it's a co-injection. There are a lot of caveats with this, but that's what we went after, just to start this mechanism. And what we found was, cancer cells are floating, aggregates or not, spheroids or not. It doesn't matter in this case in the peritoneal cavity. And there's macrophage in the beginning, before the first treatment, and then there's the omentum and other organs, visceral organs in the cavity. After you inject beta glucan particles, like I said, they're dead yeast, just like these shapes. Then the macrophage phagocytoses them and activate the clotting pathway, and then actually, trapped or aggregated these cancer cells, as well as, some neutrophil-derived factors, extracellular traps, nets that are contributing to the other half of the clearing of ascites. And this is kind of an unexpected finding, a new finding from, different from what we discovered in the past in the infection model. So I'm going to show you, especially again to the students in the audience, how we discovered this very briefly, very using a few key data sets, and then for students, in order to draw a cartoon like draw a model like this, so you have to test them. So the way to do this is you have to come up with a robust readout system. So the way we did it was similar to the macrophage disappearance reaction. This is essentially, as you can see, is a tumor cell disappearance reaction mediated by beta-glucan particles and macrophages. And then what we found is we were counting cancer cells in this case. We're injecting cancer cells and beta-glucan, and 5 hours later, and then you count, how many tumor cells you can get out of the peritoneal lavage, and you don't see much at all. And across, it's the same for many cancer types. This is a readout, and once you have the readout, you can manipulate the system and knocking out this and that, and to see basically knocking out this half of the mechanism, this half of the mechanism, see whether this number goes back to normal, the cancer cells, to tease out the mechanism in this model. All right. Another thing is to do observational studies. In this case, we were really happy to see these millimeter size as what we saw in the past infection models. These clots, they are green because cancer cells are having GFP, and we did flow, there are cancer cells there. You see them, that's suggesting they're clotting or aggregating. And then the other thing we did was we looked at momentum, because I will tell you why. Because this half didn't rescue the whole thing. This is the momentum in mouse, very hard to see. However, after beta-glucan injection together with cancer cells, you can see a lot of green signals that are there. So how we did this was, this is just one piece of the evidence, because NETs and Dectin-1, SIG, we did all of those using knockout mice mostly. But here is just the data you will see for other, genetic manipulations as well. What we did was looking at cancer cells in the peritoneal lavage, beta-glucan injection, they're gone. And if you block coagulation and adhesion, as we did in the past, it only reversed about 50%, half of it. And we were wondering what's happening, what's the remaining. Then eventually, like I showed you, omentum was contributing to this. We did omentectomy to remove omentum. As you can see, if you remove both omentum, and the clotting pathway, you reverse 100%, really beautifully demonstrating it is the omentum. And we did the same thing with Dectin-1, SIC, and PAD4 knockout mice. It's beautifully, reproducing this, phenocopying this. It was just really satisfying. I'm sure Brenna, who's on the call, agreed what I said. It was really satisfying moment for her as well. All right. Now we know we got rid of this part, and then what happens to the immunosuppressive microenvironment, which is really important for many cancers. And the reason we had to do this, I'm going to show you why, is, we did beta-glucan treatment in the beginning, as you can see over here, beta-glucan alone. And yes, the ascites was gone. However, the tumor burden using luciferase or bioluminescence or tumor burden, tumor weight, metastasis, nodules, scores, it didn't really change much with beta-glucan alone. We did so many mice that you can see there's no phenotype. We tried really hard. Thanks to the beautiful work from, Dr. Mikola Ekblat's lab, the cancer cell study using LPS and MPLA and interferon gamma in 2021, we kind of borrowed the idea, maybe we should do a combination therapy. That's what we did, and beautifully, this is July 2022, I think, more than half a year after I started my lab. I was really happy. I was the first person to see this data, and I was really happy to see this. So there, as I said, beta-glucan treated, there's no ascites, but tumor burden was also smaller based on multiple type of readouts. This is just one of them. All right, to get into the key mechanism, the novelty of this work is we're trying to understand how does the anti-tumor immunity work after beta-glucan interferon gamma, and what are the roles of macrophage myeloid cells. I haven't been talking as much, besides their clotting mechanism. So we did single cell, just like analysis, macrophage monocytes, just like every single smart immunologist would do. And we found multiple clusters as always. And what's interesting was, okay, we're looking for a change that was specific to the combination treatment, just phenocopying the tumor, consistent with the tumor phenotype. And as you can see, we luckily, honestly, really luckily, we saw this expansion of this cluster and reduction of this cluster, specifically in this double knockout. I mean, combined treatment treated group. And then there's other subsets that are also changing, especially this cluster 8 is gone in beta-glucan treated as well as combined. So that ended up being the the large max, the peritoneal macrophages. And by the way, this is a tumor, omentum tumor, single cell, not the peritoneal lavage. We also did that. That was not as helpful now as a tumor, and we focus on the tumor. So these beta-glucan treatment not only get rid of the fluid resident macrophages, also over time also got rid of the tumor adhered large max, which was another study I didn't cite in this, I didn't mention in this talk. They activate inflammasome activation and others that killed them. All right, so what are these subsets? What are the differences? I tried a lot of analysis. Eventually, again, very lucky. So we focus on cytokines and using ingenuity pathway analysis. We compared class 2 versus class 1, and then just very luckily, we're really happy to see some interesting cytokines showing up that are enriched in this class 2 that are interleukin 27 as a really good, I guess a pretty decent, a pretty good myeloid cell immunologist. I've never heard of IL-27 back then in 2022, and that's why I was really excited, and so did the first author of the paper, Brenna. And we followed up on IL-27. What is IL-27? First question. It's IL-6, IL-12 family cytokine. Thankfully, Dr. Chris Hunter, who is on UPenn campus, pioneered the work of 27, and that's where we learned a lot from him, also collaborating with him as well. So the reason why it's considered IL-6 family cytokine is because of the receptor IL-27 is GP1 of the heterodimer receptor is GP130, which is shared by all IL-6 receptors, family cytokine receptors. And also the reason why it's called IL-12 family cytokine is because the heterodimer of IL-27, P28 and EBI3, they are very similar to P35 and P40 and other IL-12 family cytokines. That's IL-27. And what's more interesting and what's more complicated, also interesting to me, is it has a dual function that was eventually, again, pioneered by Chris and others in the field. In the beginning, because it was IL-12 family cytokine in the beginning, they know it's very potent in activating T cell, interferon gamma, CD4, CD8, NK cells. That's all good. Eventually, they also found, knocking out F27 signaling by, knocking out receptor or the heterodimer P28. They saw some interesting phenotypes of these T cells in the infection models. They were actually, they turns out IL-27 can inhibit these cytokines as well. Maybe also through regulatory T cells, which, as you know, won a Nobel Prize a few days ago. Then that's what's really exciting and interesting to me. So as you would predict in tumors, since it has dual function in tumors, it's also very messy, which is stuff I like about. And then it can be pro-tumor, anti-tumor, can do all these things. As I mentioned, the CD8 activation. However, Treg expansion, survival also important. So different tumors, they're different. As you would predict, there's multiple papers showing, including a recent 2025 Nature paper, showing it's anti-tumor. However, there are studies, also very, very good publications from prestigious labs and PIs showing IL-27 can potentially promote tumor growth in different tumor models. So what happens to our ovarian cancer model in this beta-glucan and from gamma background? We neutralized IL-27 in that paper. We found, okay, that's pretty good. Actually reverse this anti-tumor response. Eventually it was partial response, but we were still happy about it. In omentum, tumor burden, mesentery, metastasis, it's all reversing the anti-tumor response, really suggesting IL-27 in ovarian cancer is likely to be an anti-tumor in this model, in these models at least. And we also did a survivor, which is probably not as important for this audience, but using different models in ovarian cancer patients from ovarian cancer patients, homologous recombinant deficient, proficient, which is the worst, has a worse prognosis in ovarian cancer patients, CCNE overexpression. And it turns out our treatment has in some mice has a curative, potentially curative, response. And the combination in this BRCA deficient mouse model is really, impressive. What about in humans? And we did a very, I guess, preliminary human analysis. We include in the paper. We analyzed these, ovarian cancer, patient-derived monocyte macrophage myeloid cells, and we identify IL-27 and EBI3 cluster in this. Interestingly, the CXCL10 co-expressing subset and CXCL10 and 9, these are potent anti-tumor, cytokines and important for immune checkpoint therapy. So luckily and interestingly, in our mouse model and also in this human data set, it's co-expressed with these macrophage subset. I don't know why it's called M2, but they're potentially anti-tumor. And also correlational study, like many good cancer biologists, which I'm trying to become. I also do correlational studies in metastatic ovarian cancer patients. Higher expression of 27 EBI3 correlates significantly with a better survival. All right. So the remaining question for this part is, basically the projects I'm working on right now, and then hopefully I can report in the next few years. And these are the stuff I propose in my grants and NIH grants. And how can we safely translate this therapy into patients, as we know is highly toxic, side effects, systemic inflammation, interferon gamma, and beta glucan is also having its own big problem. We're trying to develop a way to translate them into patients, or lower the toxicity. Also, is this IL-27 positive macrophage is a really novel subset or a new subset we can leverage? Like, can we understand how they were, developed, and what are the molecular transcriptional mechanism that are regulating the development of these macrophages in the absence of beta glucan and interferon gamma? Can we expand them? Can we develop cell therapy? All these things are the things that we're working on right now in the lab, and also. We are really interested in the IL-27 signaling. How does it work in the ovarian cancer setting? The mechanism could be very different from what's been known, what's published, and we have some hint right now. Hopefully, that can be, finished not far. All right. Next, probably 15 minutes, I will talk about a project that's not published yet, was under review. Hopefully, will be published very soon. The question we care about is acquired chemo resistance in ovarian cancer. As you can see, it's just one of the readout of CA125, a marker for ovarian cancer. And then most patients, which is interesting to ovarian cancer patients, most patients has 70 to 80%. They actually respond to chemotherapy right away. And over time, you keep treating them, and eventually they become, basically it's a selection from a cancer biology standpoint. Eventually they become, chemo resistant, and then, it's really bad. We're trying to understand the acquired chemo resistance. How do we do this? And the way the first author actually drove this project, titled Miyamoto, who is back in Japan, a physician scientist in Kyoto University. What he decided in the beginning was to compare, published data set pre and post chemotherapy, like paired analysis and RNA sequencing, to start with RNA sequencing. And then because we are a myeloid cell lab and we want to focus on myeloid cell derived factors. So, for example, cytokines, somehow we discovered, we stumbled upon IL-27. So we were just, okay, let's try another cytokine. Then, really interestingly, also luckily, we found, between pre and post chemotherapy treated patient samples, RNA sequencing, published data set, we identified that using IPA again, focusing on cytokines, we can see that interleukin-1 beta was one of the most highly enriched signals in post chemo treated samples. And also he did titled more analysis using patient published data sets, and really interestingly, in chemo-sensitive patients who respond to therapy, there was not really a change in IL-1 beta expression. Looking at this molecule, this is looking at the pathway, and then in chemo-resistant patients, they actually went up. And then we also looked at the key inflammasome, which is the secretion pathway for IL-1 beta. They are similar as well, which is really promising. And we also looked at IL-1 beta survival in metastatic ovarian cancer patients. So in contrast to IL-27, it's correlating with worse prognosis. And also we analyzed the public database, which is the same paper as I cited just now in IL-27 study. We were trying to understand what cells are secreting IL-1 beta in humans. And this subset is myeloid cells, mostly macrophages, because there's no neutrophils because technical issues. And these cancer cells, they don't secrete as much compared to myeloid cells. Fibroblasts not doing much, T cells not much. All right, can we study this in mice? Can we find a causal relationship between IL-1 beta and chemo resistance, in mouse? First, we trying to identify a chemo-resistant ovarian cancer model. And just like in patients, we do the, paclitaxel together with carboplatin, which is standard of care treatment in ovarian cancer patients. Luckily, we had access to these cells from Dr. Weinberg and Pepin's lab, and they are resistant to this treatment regime, which is the same as resistant, patients. And we also did a single cell RNA sequencing on this just to confirm, okay, IL-1 beta is also coming from myeloid cells. In this case, this is macrophages, a little bit DC, a lot of neutrophils, which was missing in human data set. And we also confirmed that because a lot of people are saying cancer cells secrete IL-1 beta driving this and that. We confirm in our model cancer cell not secreting much at all with or without stimulation of chemotherapy, and LPS treated macrophage as a positive control, showing that it's highly, it's the experiment worked. And what do we do to test whether it's causal or not? First thing to do is knock it out. Let's delete it and see what happens. This is the data I'm showing you here. These mice are all treated with chemotherapy, and as you can see that using different measurements of tumor burden, and which is a standard in the lab right now, bioluminescence, tumor weight, metastasis, and digest the tumor, count tumor numbers, and we also do section as well. As you can see, IL-1 beta knockout mice develop much less smaller tumor after chemotherapy. And more importantly, without chemotherapy, IL-1 beta did not have any difference, tumor growth difference between the wild type, suggesting the chemotherapy-induced chemotherapy-dependent manner, which is the same as in patients, which is really good. And to rule out the direct, role of IL-1 beta on cancer cells, again, a lot of literature on this. I want to make sure that's not the case. In our models, we use KPCA cells. I think we also use other cells, incubate KPCA cells together with IL-1 beta with different concentrations, and test their IC52 chemo drugs, carboplatin, paclitaxel. As you can see, nothing changed. There's no significant difference in this model, suggesting IL-1 beta is probably not directly, killing tumor cells. Then how does it work? So we look at tumor microenvironment. In this case, to begin with, we always look at flow cytometry because it's easier and cheaper, and you can do it anytime. As you can see, we found a very specific, interesting phenotype to neutrophils. You can see that in the tumor, neutrophil number percentage and numbers goes up. This is the percentage I'm only showing you, but numbers also do the same. And in wild type mice, however, if you knock out IL-1 beta, neutrophil number didn't go up. Also in the untreated, it just didn't change, suggesting is this potentially the reason why it's correlating with the tumor phenotype, right? So is neutrophil the case? Other cells not changing at least significantly? What we did was we depleted neutrophils, as many others have done. So what we did was we used this neutrophil depletion protocol. I think I was introduced by Filippo Viglia, who was a postdoc with Dimitri, who's back in Wistar now. He told me to use this newer protocol, which is more potent, anti-Like6G together with anti-rat IgG, basically make a extended half-life extended Like6G and also multivalent. So the method was published a few years ago in Nature Com. Apologize, I forgot the last author. And it's really good. We did the same thing, as you can see, in chemo-treated Wild-type mice depleting neutrophils, tumor burden got less, suggesting neutrophil is mediating some of the chemo resistance. And however, in IL-1 beta deficient mice, you don't see this change after depleting neutrophil, suggesting they are likely to be in the same pathway. Of course, it's not definitive, but a good suggestion. And mesenteric metastasis showing the same thing. We did single cell sequencing flow analysis. Okay, we found the effector T cells in IL-1 beta knockout mice is doing more, is getting more enriched and consistent with other markers, which is not surprising because we know neutrophil MDSCs, they are suppressing T cell function. In this case, they have less MDSCs, neutrophils just suppress less. So it was not, I mean, honestly, not mind-boggling to me at all. That's why we were not happy, we're not satisfied by this. And then we were asking, okay, what other ways can neutrophil contribute to chemo resistance? And luckily, or interestingly, like I think a couple years ago, 2023, this paper came up showing neutrophil nets. Basically, it's promoting chemo resistance, cancer cell specific, cancer cell intrinsic manner. And then we look at nets, stand, citrulinated histone H3. You can see that in wild type treated with chemo, there are a lot of nets, and wild type knockout mice don't have much chemo, don't have much nets at all. And we did knockout mice from our collaborator at Wistar, Yulia, and Nefedova, and nets knockout is showing a lower tumor burden, consistent with IL-1 beta knockout. And we also did in vitro, just like the paper showed, which is not as impressive, but is showing the same thing. Not as impressive as the paper, the cancer cell paper. Essentially, if you treat them with chemo and put nets, supernatant, no neutrophil, and then a tumor grow faster, and then if you inhibit nets with GSK inhibitor, the tumor get killed fast, suggesting nets is likely contributing directly to cancer cell intrinsic chemo resistance. Then the question we were asking was, okay, what cells are mediating this? Where I personally is really interested in the cellular crosstalk of immune regulation. And then we were asking what cells expressing IL-1 receptor, IL-1R1. And we did single cell sequencing, also together with published data set in humans. We can see this subset of cells that are turn out to be fibroblasts that are expressing high levels of IL-1 beta receptor, IL-1R1. Same thing for our mouse model. There's some cancer cells expressing it, but different patients have different expressing levels. It's not consistent, not the highest. In mice, it's the same. This is a fibroblast, two subsets of fibroblast, progenitors, I think normal fibroblast. I'm trying to learn more, which is really complicated, just like macrophages. Cancer cells and mesothelial cells, they express some, but not much. And with the flow analysis, protein level, yes, it confirmed the fibroblast, at least a subset of it, express, just like in humans, express high level of IL-1R1. And what we did, to finish up this story, was we knocked out IL-1R1 in the host, knockout mice, not tumor cells, and we saw consistent tumor, chemo sensitivity in IL-1R1 knockout mice, suggesting it's, confirming our model, which I will show in a second. And same thing, the neutrophil phenotype is consistent in IL-1R1, not other cell types. And we also look at neutrophil chemokines that are recruiting neutrophil. The classical ones are CXCL1, CXCL2 in mice. It's after knocking out IL-1 beta, you can see it's going up, suggesting these are, in fibroblasts specifically, these are likely contributing to the difference we saw just now. And also we analyze human data set, all these neutrophil, in humans are more complicated, CXL2, and most important CXL8 is IL-8 is increased in post-chemo pair samples, I think in this case as well, in multiple data sets. And lastly, IL-1 beta is correlating with more neutrophil infiltration in ovarian tumor patients, IL-1R1, CXL2, not surprising, but these two are interesting to know. And lastly, we also did human analysis, which like section analysis, which I'm not showing you here. But basically, we think we found that in ovarian cancer patients in this during chemotherapy treatment, so chemotherapy is driving infiltration of IL-1 beta positive cells, and then secreting IL-1 beta, activating fibroblasts, secreting CXCL1, 2, recruiting a bunch of immunosuppressive nets, releasing neutrophils that are suppressing T cell function and inducing cancer cell intrinsic chemoresistance. One of the big problem we have, or like question we have for this part, is we couldn't figure out. That was different from published literature, is what's driving the IL-1 beta secretion. So we try co-culture and this kind of things. Maybe we should try more, but for now we still don't know what's inducing IL-1 beta secreting cells in the beginning before neutrophil comes in. Is it macrophage, monocyte? We don't really know in the beginning. All right, lastly, just acknowledging people who did the work, and Tito and Brenna. Brenna started the first project, IL-27 project. Tito started the second project, and helping Brenna finishing the first project. And UJ came along last year, helping Tito, who left, went back to Kyoto to finish up this IL-1 beta story. This is the lab, and I already explained, and people who contribute to the project and the funding. And thank you very much for your attention.
Thank you very much for a fantastic, informative seminar. So many new concepts to consider. We have some questions in the chat, but I'd like to invite the people who put their questions to speak up and ask their questions directly. First would be Hironara Akasaka. Are you still here? And would you like to ask your question? You can unmute, and he's here. Or she. Yes. Okay. I'll ask the question. Or if you can read it.
Yeah, I got it.
Okay, okay.
Oh, sorry. Go ahead. Go ahead.
Okay. Have you examined whether similar macrophage aggregation or clearance responses can be induced in human peritoneal or, sorry, I just moved, acidic macrophages, given the known interspecies differences in macrophage phenotypes? You may have addressed this, but go ahead.
Yeah, so we get this question all the time. I didn't do it because I don't know, and we did in vitro work, which was unpublished. I think they do in vitro also same as mouse peritoneal macrophages. They put them together in a dish. You don't want to dilute the coagulation factors too much. You put beta-glucan, zymozyme in, they would aggregate as well. But we didn't publish it, and then we're hoping to translate into patients. This is the one thing I didn't mention, Brenna's idea, and then we're trying to, just haven't got money for it. Leverage beta-glucans clotting aggregation, to eliminate ascites, which is a big problem, alone without interferon gamma. And we're exploring nanoparticles and others, but we haven't really, take a serious stab at it.
So I have a related question. Ascites includes an increase in cells, but also fluid. Is there any change in the amount of fluid that is released when you treat with the beta-glucans? Or
yes, good reviewer question, and we addressed it. And yes, we quantified the volume. The volume I showed you in the picture. The reason why it's large is because it's a PBS wash. It's not true ascites volume, and the model we used to have different amount of ascites, and then eventually we showed that yes, it also reduced the absolute ascites volume.
Okay, and is the ascites volume due to vascular leak?
Yeah, it's also a good question. We get it a lot. So there are multiple theories. One theory I like is, it's always about leakage and the drainage. Lymphatics, Dr. Go-Young Ko from Korea and others show that lymphatic drainage was blocked in the diaphragm. There's subdiaphragmatic lymphatic drainage system that's blocked by, actually we saw this aggregation as well. Again, it's in my K99, but it was never published. And so we saw the adhesion of macrophages. They saw as well. After tumor and after beta-glucan treatment, they would block the drainage, and the leakage clearly is a tumor phenotype. It's across many tumor types.
Okay, thank you. Let's see. So Arthur Dyer, would you like to ask a question? Yes.
Yes, sure. Thank you for the talks. I found them really, really interesting. My camera's off, sorry. I had two questions that I snuck in, one of which was if there's any indication about trained immunity, because beta-glucans, like there's a lot of kind of literature on that. And the other one was, so CLEC7A is kind of the traditional way that beta glucans are going to signal. Do you find it interesting that it's kind of come through IL-27 instead of CLEC7A?
Yes, we tested the Dectin-1. Honestly, clearly, I mean, two questions. Training immunity reviewer asked, and we honestly we shied away from it because but reviewer two insisted it. We did a preliminary analysis, put it in the paper. It was at best conflicting a little bit. So we did beta glucan treatment, single treatment, combined treatment, look at bone marrow cell numbers based on their protocol. And we somehow did not see a significant change after beta glucan alone treatment, which was shown by many other people. Maybe the protocol they told us to cite was not the right protocol. It's a week post injection, which maybe was not enough, but it was published by some people. Then, however, we did see an increase in hematopoiesis, lots of myeloid cell progenitors in the combined treatment treated mice, beta glucan plus interferon gamma treated mice, GMP, MPP, all those things. And that's for the training immunity. We did not test, we did not go deeper than that because I feel like it's been sort of well established in the field, and I don't know how much I can contribute honestly. That's why I went after the IL-27, which was less well known. CLEC7A, which is Dectin-1 in mice. Surprisingly, Dectin-1 knockout mice did not matter. We don't know why. It's not like IL-27 you reverse or whatever. That does not matter. So that's why we're trying to understand how there's a project in the lab really understand how beta glucan plus interferon gamma it's really working, and we're finding something unexpected, which is always something I love, so I don't know yet. I don't know why it's not doing it, but I know the clotting is dependent on tissue factor, which is expressed on the macrophage surface. We tested in the previous paper, and I think. Again, in proposing my K99 it's because of phagocytosis of these particles that's messing up the flippage, floppage on the membrane, which is critical for tissue factor activation, decryption of tissue factor. That's activating the clotting. And the hidden part, I don't know, but maybe the tail end part is also connected, but I don't really know the details. But good question, thank you.
Okay, thank you. From Fabio Qualia, are you here?
Qualia, yes, thank you. I was wondering if you try to remove T cells and you still see the effects from beta glucans, the clearance of the tumors. I don't know, in Prime also showed that T cells activation was important for their clinical trial, and I don't know, maybe also if you had the immunocheckpoint or you explored that as well.
Great questions, really good questions. We did all of those. Some of them was in paper, some of them not, because the data was negative. And then, so the data we put in the paper was T cell dependence. Yeah, so that's also interesting in a way that's different from traditional training immunity. Because training immunity, traditionally speaking, it's mostly macrophage neutrophil phenotype and independent of T cells. A lot of papers on that. But we deplete T cells and the tumor phenotype was gone. That's why a tumor control phenotype was gone. So tumor just was growing really bad in beta glucan treated mice, beta glucan plus interferon gamma treated mice, really suggesting it's a crosstalk between macrophage and maybe something else we're trying to identify, activating on T cells, and that's killing tumors. And then the ICB, the checkpoint thing, that was the first one of the first baffling, mind-boggling data we got. It was making it worse. This was so basically combining PD1, and we saw okay, PD1 is having a problem. We tried PD-L1 with CTLA4. Then it's doing the same thing. Together with beta glucan plus interferon gamma, it made the tumor worse, did not make it better. That's why we combined with carboplatin in the paper eventually. The reason why that was the case was at the time when we had that phenotype, a paper from Dr. I forgot his first name, Dr. Zhou from Michigan, a very established immune checkpoint PI. And then he published cancer cell paper showing there's these hyper progressive patients who to immune checkpoint therapy. The reason why they were hyper progressive was because the high interferon gamma signature. So I was thinking, oh, okay, that kind of explains why we're making it worse, because we're giving interferon gamma, and that kind of came back as a beautiful circle, though we did not follow up on that.
Thank you.
Okay, Minglu Yan.
Okay, great.
Great.
So do you have any thought about which signals could possibly induce the L27 expression upregulation in the specific macrophage subset?
These are great questions. All of the questions, yes, are really good. And where some of the questions we haven't addressed, like this one, we're actually working on it. This is one of the aims in my R1, and was doing it. I can just the preliminary data. Okay, the preliminary data is suggesting it's likely lysosome activity. Like I said, phagocytosis. When I was mentioning just now, I was suspecting it for a long time. Eventually, luckily, in the past summer, we had a summer student from Penn who is still with us, and then she did a lot of qPCR analysis. We think autophagy or vesicle trafficking, phagocytosis related pathways are controlling IL-27 induction. Not only IL-27, the whole signature genes of that subset of macrophages, MHC class 2 and other things are all dependent on lysosome signaling or phagocytosis. We don't know what else is doing it, and then hopefully I can get the money and tell you in a few years.
Terrific. So I have a question about the basic discoveries that the resident macrophages disappear when you induce inflammation in the peritoneum. So what do you think is happening given all of your results? I think you've showed before that the macrophages clot and precipitate, but what do they do once? Eventually, if you remove the inflammatory stimulus, they come back. So what is the fate of the macrophages that precipitate? Are they dying? Are they?
Yeah, really good question, Judy. And I guess explains why my K999 was well received. I didn't know it was that important, but I'm glad people are concurring with this. So the fate of these macrophages partly was answered by the immunity paper from Carlos in Spain. So they did follow-up study, which I also saw, is the inflammasome activation in the macrophages, resident peritoneal macrophages right after phagocytosis of E. coli or zymosan particles. They activate inflammasome. As he saw in the paper, I saw it as well. ASC specs. So he basically attributed this to inflammasome, the disappearance, the death of these resident macrophages. However, it really depends on the dose you use. What I proposed was to use linear tracing fate mapping experiment to follow up, whether how do they come back? Do they come from the resident pool, or do they come from the monocyte pool? It was always a fun question to ask as a macrophage biologist. And I did preliminary analysis using TD tomato positive, those fate mapping models, and then depending on the dose you gave. Another paper from I think Phil Taylor at UK and Caleb Bain, they showed that different doses of diamazan will induce different infiltration. I mean, which makes sense because different inflammation, right? Different degrees of inflammation would induce different infiltration of monocytes. So that's what we found as well. If you give a 1 mg, I mean, 1 mg, all of the cells will be gone, and then most of them will be coming from monocytes. Low dose, 10 microgram, they also induce clotting, but they bounce back really quickly. That's coming from, crawling out of the clots. that's from resident pool. Yeah, it's a kind of long answer, but it's a really good question. Really interesting question we were thinking about.
So do you have any evidence that there's like a resident progenitor that might repopulate?
Oh God, love that question. Nobody knows. No, the answer is nobody knows. I think there's more work now, because I'm not really working specifically on that right now. Like I think it was a bioarchive paper I saw like last week from, I didn't read the detail, from Jie Song's lab Jie Song's lab from Virginia in alveolar macrophages. I think he was trying to study the stemness of alveolar macrophages. And I think yes, I agree with you. There's a subset of macrophage in the peritoneal cavity. They express c-KIT. They express, they're proliferative. I don't know how much they're contributing, but in a steady state, yes, they maintain, as Florent mouse model, MS43, monocyte fate mapping. We see it, we reproduce, they saw it. It's without perturbation. The monocyte pool would remain at 40% throughout maybe life or something. But we now care about the inflammation, in cancer, and then that we're doing that. That's in different treatment. That's gonna change. But in the steady state, I think you're right. It's probably the stem cell pool there, and nobody has reported yet.
Okay, terrific. Well, are there any other questions that anybody would like to directly ask? This has been a fantastic seminar. I actually have some observations in ovarian cancer I would love to collaborate with you on.
Yeah, we'd love to collaborate as well. Yes.
Yeah. So let's get together soon and talk about it. Yeah.
Yeah.
Okay. Well, wonderful. Thank you, everybody.
Thank you.
Thank you, Nan,
everyone.
for a great talk.
Thank you. Thank you, everyone. Thank you for inviting again. Yes. All right. See you soon. Bye-bye.
Bye.